Hello, everyone, and welcome to today's webinar, Early Identification of Target Candidates for Pancreatic Adenocarcinoma Immunotherapy. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotec. To learn more, visit miltenyibiotec.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker, Dr. Olaf Hardt, Senior Manager of Research and Development, Miltenyi Biotec. Olaf, you may now begin your presentation.
Thank you for joining my presentation today about the early identification of target candidates for pancreatic adenocarcinoma immunotherapy. As probably all of you are aware, CAR T-cell therapies really have gained a lot of interest because of their tremendous efficacies in treating, in particular, hematopoietic malignancies over the last years. A very prominent example being ALL, where now also CD19-directed CAR T-cells have gained FDA approval recently and really show a nice example of cellular immunotherapy efficacy. However, so far, this success is really limited to hematopoietic malignancies, such as ALL and some others, whereas there's very limited success in treating solid tumors, and that has a range of reasons, such as a very immunosuppressive immune microenvironment, but also the lack of known target structures, which you have, for example, here in ALL. There's the lineage marker CD19 that is expressed on almost all of the tumor cells in many of or most of the patients, but this is not the case when you have solid tumors. And in particular, also these healthy cells, which are also targeted because they express CD19, are actually dispensable, meaning the CAR T cells that eradicate the tumor cells and also eradicate the healthy B cells is still an option, because you can do this by a therapy intervention that can still be tolerated well by the patients. And that is not the case in many solid tumors, because the structures that are expressed on these solid tumor epithelial cells are also expressed on cells of other healthy organs that are not dispensable. So let's briefly go into the principle of CAR T-cell therapies. So this here is shown for an autologous setting, where the patient is actually undergoing a leukapheresis. The T cells are separated from the leukapheresis and are then ex vivo modified, genetically engineered to express the CAR or the chimeric antigen receptor on the surface. This is mostly done by lentivirus, but also mRNA and other methods can be applied. Afterwards, the cells are further activated and expanded to huge amounts, which are then reinfused back into the patient, where the CAR T cells can efficiently migrate to the tumor cells and then kill them by recognition of the cell surface molecule expressed on the cancer cell by the CAR, and then afterwards cytokine release and also active killing of the cancer cells. And on the right-hand side, you can also see a little bit the differences between a T-cell receptor-mediated and CAR-mediated targeting. So both have advantages and disadvantages. For example, the CAR is very efficient in treating cell surface marker-based approaches, because it is not restricted to MHC molecules, so it's independent on the haplotype of the patient. So basically the same targets can be applied that you can also apply by biologics such as antibodies or BiTEs. However, the disadvantage is, of course, that you can only target cell surface structures, whereas for T-cell receptors, you can also target intracellular structures and even mutated antigens, because they are presented in MHC fashion. So today I will focus purely on CAR T cells. And as mentioned, one of the major bottlenecks is the strategy for identification of the best therapeutic targets, which have to be cell surface targets. And we, over the last couple of years, have really worked a lot on this for different tumor indications. Today, as mentioned, the focus is on pancreatic ductal adenocarcinoma. And we have developed this strategy here, where we start with solid tissues, either from patient-derived mouse models, xenograft models, or from primary human specimens. And as Jana already pointed out, did this here by high throughput screening of cell surface antigens by flow cytometry in order to narrow down the amount of markers that are of interest, and then do further spatial identification of these marker candidates, let's say, in the tissue context. And this leads to the selection of top candidates for further downstream functional evaluation. So let's go into this a little bit more in deep, because here you see also a little bit more the initial early workflow, where we started screening of about 400 antibodies for binding on pancreatic tumor cells. And therefore, we have also used 19 different patient-derived xenograft models. And this has the advantage that here, tumor material is not limiting, meaning that you can easily do flow cytometry-based screenings and do parallel analysis of single samples per screening antibody. Whereas in the second course, when you then want to go more into the validation mode, and you want to do this on primary tumor, but also in particular on healthy tissue specimens in order to identify potential on-target of tumor toxicities, really the tissue becomes the limiting factor. And here we have done sequential screening by immunofluorescence, and I will also briefly talk about this later. But first of all, let's talk about the screening by flow cytometry. So here again, we splitted this up. As mentioned, we started with PDX model, having hundreds of millions of cells. And then we applied these screening plates that we provide, the MACS screening plates, and then also narrowed the marker number down to a more narrow data set, and then also screened many more PDX models again. And this all was done by flow cytometry. And then after narrowing down the marker of candidates, we have also worked even with flow cytometry with primary tissue specimens, which were coming from surgical resections of primary tumors, and then have also stained them by flow cytometry. So of course, when you want to do flow cytometry-based screening, you have to have a very efficient way to get a single cell suspension. And that can be very tough from certain tumor types, such as pancreatic cancer, which usually is very desmoplastic and very tough. And so basically you have two goals. The first goal is very obvious. You want to disrupt the extracellular matrix and cell adhesion components without destroying the integrity of your cell membrane. And for this, enzymatic digestion is usually the most efficient. However, second goal that is often not really well considered is that you want to preserve your cell surface target candidates, of course, because if you lose a lot of these markers just by using crude or aggressive proteases in the first step, then you would also have a lot of false negatives that you for sure want to avoid. And for this, mechanical disruption usually is the method of choice. However, many cells which are really well associated in the tissue, such as the epithelial tumor cells, cannot be derived efficiently only by mechanical dissociation. Usually, and most efficient is a combination of both the mechanical and enzymatic treatment. And for this, already many years ago, we have developed optimized solutions in our tumor dissociation kits for human, but also for mouse, where you can get actually very nice single cell suspensions of highly viable tumor cells, and also all the other compartments that you have in the tumor. And these cells are also show improved functionality. And according to the second goal, I mentioned the preservation of key cell surface antigens. We could actually show by screening more than 200 epitopes that we've tested on human and mouse tumors upon dissociation, that only about 5% of these markers are affected by our enzyme combination. If you compare this to very aggressive enzymes, to trypsin or dispase, usually you see then more than 50% are affected. And because we are using highly purified enzymes and a mixture of certain enzymes, we can exactly control the reactivity, let's say, of these enzymes. And we know that these about 5% sensitivities come from enzyme R. So if you reduce this, you can preserve all the markers that you have. So you can refer to these lists, can see if your marker of choice is sensitive or not. So you do not have to do this empirically in-house. And then you can follow this up, reduce enzyme R if there's a marker actually you're interested in and that you want to analyze. And this is the data from these initial screenings. And for the PDX models, this is just a snapshot because I mentioned we screened many more markers. So this is just exemplary data. So what you want to actually get here is you only have human tumor cells, of course, because all the other cells are mouse tissue. So you can go for on-tumor binding, but you cannot evaluate off-tumor binding, of course. So in the first screening, we really go for the specificity on the tumor and choose markers that are highly expressed on the human tumor cells in this setting. And then secondly, when we go to primary human tissues, you can then, of course, map the marker expression on your tumor cells, but you can also compare it to the expression, for example, on immune cells and other cells that are still present in the tumor tissue. And by this then, together with bioinformatic safety ranking, you can come to a more close number of markers that are serving as potential target candidates. So then that brings you to the second step, where you maybe want to then further analyze and validate your target candidates on primary adenocarcinomas, but also on normal adjacent tissue and healthy tissue microarrays. And as mentioned here, the tissue specimens are really the limiting factor. So you have to go for another method. And that is usually immunofluorescence, multiplex immunofluorescence. However, if you compare standard microscopy, which easily can be done in 5-plex or even some more, but that has to be tremendously validated. However, in this 5-plex, usually you cannot then always screen 5 target candidates, because you also want to have an idea about the lineages that you have. So you usually stain also immune cells and tumor cells to see really the co-expression of your marker of interest. So if you want to stain 100 markers, you have to do 30 to 50 serial sections. And as each section is really similar, but still a little bit different, you completely lose then the single cell information. And that is in particular important if you want to do correlation analysis, which targets are co-expressed on which cells. And for this, we have developed the MACSima technology, and that is a cyclic immunofluorescence staining method, where you can really go until several hundred plex marker analysis on the very same tissue section, so keeping the single cell resolution, of course. And by this, you can then, of course, also apply the lineage markers and all of your target candidates in one run and all stain them on the very same tissue section. And this is just one example here from the markers that were interesting from the flow cytometry. What you see here in a primary pancreatic ductal adenocarcinoma, and just to focus on a few here, you see, of course, that some are tumor-specific, like CD318, where you see really a nice correlation of the epithelial cytokeratin signal and our marker of interest, whereas others, as expected, like CD59, are not tumor-specific. So they are expressed on tumor cells, but also on stromal cells and endothelial cells. So you can then exclude such markers from further analysis. And this, of course, is very helpful on the tumor tissue already. However, also you want to see the potential on target of tumor toxicity in other organs. And therefore, you can then also use healthy tissue microarrays like here, where you see again CD318 being nicely expressed on pancreatic cancer, but not on the other tissues. There's a slight background expression in the colon, but this is mostly in the mucous region. Whereas, for example, for TSPAN8, you see a strong expression in the pancreatic cancer sample, but also very strong expression in the colon. So this target for sure needs further evaluation. But actually, this is very similar for other targets such as CA, which are already under clinical evaluation. And there are other methods to overcome potential toxicity, such as different routes of injection or safety switches within the CAR T cells. So then upon having enough validation and confidence in your target candidates, you can go on for functional evaluation. And first of all, you have to then clone this into an array of CAR constructs, because still so far it is not possible to purely predict what CAR configuration would be optimal for your target candidate. So of course you can talk about the signaling domains, the transmembrane domains, but one very particular case is also the spacer domain, because for many binders, you do not know where your epitope of binding is located. Is it very membrane proximal or is it very membrane distal? And in order to resemble more or less the immunological synapse, you need to really get the right spacer length in order to target shorter or longer targets. And therefore, we have done so. We have cloned SCFVs and put them into the CAR context, and then did functional analysis. And this is just one example how this works. So we have transduced T cells with our CAR constructs, and here you see a mock control, so mock transduced with an irrelevant CAR or even GFP. or also CAR T cells. And here you see GFP labeled tumor cells. And then when you add the CAR T cells or the mock T cells, and then you start the killing assay, you see that the CAR T cells very fast and efficiently eradicate the tumor cells, whereas the mock T cells cannot do so. And this can also be quantified, of course, and this you can see here. So you see that some CARs are highly efficient, some are intermediate efficient, and some are also non-functional. And next to the pure killing kinetics, of course, what is also very interesting is to get into the further CAR phenotyping. And this again is of course done by flow cytometry with checking for activation and exhaustion markers such as PD-1, but also cytokine release such as interferon gamma, but also many more. So here we're using our MACSplex flow assay to really in parallel analyze a lot of different cytokines in the supernatant of such co-cultures. So this is nice, but this is all done based on functionality on cell lines, tumor cell lines that have been cultured for decades and not really resemble the heterogeneity that you find in primary tumors. So we were wondering really, what about the efficacy on primary cells coming from patient-derived xenografts or also primary tumors? And that of course complicates the whole method, because you cannot start here where you have your T cell and tumor cell culture and then can go directly into a functional killing assay and to phenotyping, but you have to start much earlier with a solid tissue. And again, the first step would be sample preparation. I will not go into this again because we already discussed it. So, but then you have two additional steps. First of all, you have to isolate your cells of choice, so your tumor cells, and then you have to culture them. Because in many cases you might not have enough material from the beginning, so you might want to at least short-term expand the tumor cells for your functional assays, but also you have to plate them in order to do these killing assays, and that is not so trivial when you work with primary tumor cells. So let's start with the isolation of your target cells. And at Miltenyi we have developed comprehensive workflows starting from all different materials, so xenografted tumors, mouse tumors, or human tumors, and then isolate your tumor cells. As today, I will only go into xenograft and human tumors, but the principle is the same for all of them. So let's talk about the xenografts first. And here, let's say it's a more or less easy solution because you have human tumor cells in a mouse context. So what you want to do is you want to remove all the mouse cells in order to isolate your tumor cells. And therefore we have done a cell surface marker screening and have coupled an array of antibodies to paramagnetic microbeads. So the principle is that after dissociation, you have your mixture of human cells depicted here in blue and all the different mouse cells. And these antibodies, they recognize all the mouse cells from all the different tissues and all the different mouse strains. So if you run this over a column that is placed in the magnetic field, you will find your untouched human tumor cells in the negative fraction. If you want, you can also isolate your mouse cells subsequently. And this, of course, has the advantage that it's completely independent on any positive marker you need to know on your tumor cells. And therefore, it works also for all kinds of different xenografts, even for non-tumor cells. So for example, when you graft stem cells into the mouse, you can still get them back. And this is when you take the cells in culture. So if you would just culture the original fraction, what you see here is that you have tumor cells that are growing on the bed of fibroblasts and some other like macrophages. So if you would do a functional assay on this here, it would be very hard to dissect what really comes from your killing of your target cells and what comes as a bias just from side effects from contaminating cell populations. Whereas when you isolate your tumor cells, and I have to say that this whole method takes only about 20 minutes, then you start with a pure population. And of course, that would not only help you with CAR T cells, but also when you test biologicals or small molecules. And then just to make the story complete, also for the human tumors, we can isolate the tumor cells. Of course, here a little bit more difficult setting because you want to isolate human tumor cells out of the human lineage. And of course, when you ask yourself, what are the major contaminants? It's of course the red blood cells, the white blood cells, the endothelial cells, and also the fibroblasts. And again, we have developed an antibody cocktail that you can just apply. And also again, in 20 minutes, get from such a crude mixture of cells to purified tumor cells. And again, if you compare these cultures as expected from the original fraction, you have a mixture of tumor cells that are also in many cases just overgrown after very few days by fibroblasts, Whereas when you isolate your tumor cells, you can have a clean culture that you can go on for your functional assay. And this is not only possible for solid tumor samples, but also, let's say, for very challenging samples such as pleural effusion, as shown here for ovarian carcinoma, because there you usually have only 1 or 2% tumor cells, but still with the same method you can isolate such tumor cells and also make them applicable for functional downstream analysis or assays. And again, just a side note, also we do a lot of sequencing, single cell sequencing, but also bulk sequencing. And what we've also seen, but of course it can be expected, that when you isolate your tumor cells and then do a whole exome sequencing that was done in bulk here, of course you get also much higher sensitivity in the detection of somatic variants, because you isolate your tumor cells from a subpopulation to a pure population. So you're not spending a lot of reads on the cartridge, of course, on sequencing cells that you're not interested in. And the same, but even more promising and dramatic, holds true, of course, for the single cell applications. So then you have isolated your tumor cells, and then you want to culture them for the killing assay. And again, this is very challenging, but also for this, we have established a nice workflow, starting, as mentioned, dissociation, isolation of tumor cells. If you want, then you can do the QC by flow cytometry and then culture these cells, either short-term or to just plate them, or maybe a little bit longer term if you want to expand them. And here, what I want to point out is these are serum-free media. And that has the big advantage that it is shown that when you culture tumor cells in media containing high amounts of serum for prolonged times, these cells tend to differentiate and also lose their tumor-initiating capacity. And this we avoid here. So these serum-free media allow the culture of tumor cells. So far, we have optimized them for 4 indications: colon carcinoma, renal clear cell carcinoma, ovarian carcinoma, and pancreatic carcinoma. And if you compare, for example, early and late passage, you see that there's a preservation of the morphological heterogeneity. We have also done a genetic analysis and flow cytometry to really prove that this is kept. And what is also very nice when we look here now again into pancreatic carcinoma, that you can derive it from primary tumors, but also from xenografts, from unsorted and isolated tumor cells. But very nice also, you can get adherent cell cultures, but also suspension cell cultures, because we have seen a trend that many epithelial tumors tend to better grow in 3D. And I have to say that these are not embedded, so these are just purely grown as tumoroids, and our media still nicely supply them with everything they need to grow in suspension tumoroid 3D structures. And that is, of course, can be applied for more complex co-culture models. And again, what is very important, and this is data from the group of Andreas Trump, which has initially developed this media before working with us together on the further development and optimization, is that when you compare the patient originating tumor to the patient who have xenograft, you see what you expect. You nicely resemble the morphology. But what is new now to these media is that even when you culture them in 2D, as shown here, and then regraft them into a immunodeficient mouse, so a cell culture that have xenograft, still you see nicely resembling of the histological features of the original tumor. So they keep their memory of where they are coming from, and also look very similar to the PDX models. And that is, of course, giving you the chance to also work on these cells and validate everything later on these cell line-derived xenografts still. So with this now, we have all the tools in hand actually to also perform functional CAR T-cell killing assays on primary cells. And this is what we did. And here just some exemplary data. Here you can see ASPC1, which is established cell line with expressing all the 3 markers that we have talked about. But now you can also see cell lines derived from primary human tumors and also from xenografts. And now you see also there's an array of different expression patterns of our target candidates, as it would be in the reality of clinical samples. And so now you can all use these kind of primary cultures and also co-culture them with your CAR T cells and really ask yourself the question, what influences the CAR T cell activity? Is it only the marker expression? Is it a co-expression of immunosuppressive molecules such as PD-L1? And actually what you see, it's very dependent on the target expression, of course, because, for example, look at this PATU cell line here. and if I go back to the flow cytometry, you see that this cell line only expresses CD318, but not the other targets. And as expected also, when you look here for marker upregulation, but also cytokine release, only CD318 based CARs actually efficiently kill this cell line, upregulate activation markers, and also release cytokines. And for example, for the ASPC1 expressing all the targets, you see also killing of all the CAR T cells. And the same holds true for this primary derived cell line from a primary human tumor. So with this, you can then also resemble the heterogeneity in a clinical reality. And also what you see is it's not the only case, the target expression, it's also other factors, because also the cell line activation pattern is not always at the same level in each of these cell lines. So upon then really nicely characterizing your different CAR constructs, you of course choose the best working ones in vitro, and then you perform in vivo studies. And here we have used NSG xenograft models with either ASPC1 or BXPC3. And we have injected then CAR T cells. And what you see here is on this ASPC1 model that CD66s can prolong survival of the mice, but it cannot eradicate the tumor. And the same holds true also here for this BXPC3 model. I have to say that this is very challenging. Both are very challenging models because we already established tumors for almost 2 weeks. So they are solid masses before we inject IV, the CAR T cells. You see that TSPAN8, in particular with the S-spacer, shows a very nice response with tumor clearing after about 30 days. But only in the ASPC1, not in the BXPC3 model. So here you just see tumor stabilization for TSPAN8. And this again can be dedicated to the target expression evaluated by flow cytometry, also ex vivo. Here you see TSPAN8 only very low expressed on BXPC3, very highly expressed on ASPC1 cells. And our, let's say, best candidate, CD318, you see very fast and efficient tumor eradication even after less than 2 weeks in both models. So of course, to better understand why a CAR T cell is working or not, then we do ex vivo analysis. So we resect the tumor or tumor remnants, and then we check this by, again, the MACSima technology for CAR T cell infiltration, tumor remnants, macrophage infiltration, and target expression. And this is, of course, accomplished also by in-depth flow cytometry analysis, again, to look for potential explanations of the in vivo efficacy. And for example, what you can see here is that actually CD66c function was not low due to limit of target expression, because you see it's nicely expressed even ex vivo, but it was obviously a lack of functional CAR T cell infiltration that you see here in CD318, but not in 66C. So you see T cell infiltration, but not active infiltration of these purple CAR T cells here. And of course, to make the story round, we also do a lot of phenotyping on the T cells isolated ex vivo. So we dissociate different organs like the bone marrow, the spleen, but also the tumor, isolate based on CD45 positive enrichment the CAR T cells. And then we do phenotyping and quantification of the CARs, and their flow cytometry is, of course, the method of choice because of the very nice quantification you can do. But also we look at central memory versus effector phenotypes, because this was shown to have predictive value later on for also clinical efficiency when you go for translation. So last point I want to make here is that, of course, this is all either flow cytometry or 2D spatial resolution, but you might also want to go deeper in the tissue and also see the 3D context. And that is where we use light sheet microscopy with our ultra microscopes, which actually give you the opportunity. So in this case, we did the labeling even in vivo. So we injected the dyes into the mice, but you can also do it ex vivo. Then you take out the tumor or the organ of interest, you fix it, you clear it in order to make it amenable for light sheet microscopy. And now you can really see here in green the tumor cells, in orange the vasculature, and in pink you see the CAR T cell infiltration into the tumor. And you can then, of course, also make statistics about the spatial distribution of these cells, and thereby derive much more information about the persistence and proliferation of the CAR T cells inside the tumors. So with this, I want to sum up, and I hope I could convince you that we have provided a comprehensive workflow for the identification of new target candidates. Here I focus today on CAR T cells, but this is of course also applicable to other cellular therapies and also biologics. That we have developed solutions to address all the technical challenges in the functional evaluation of immunotherapy targets, in particular when working with primary cells. And finally, that we also identified 4 target candidates for cellular immunotherapy of pancreatic cancer, showing high tumor specificity, very restricted off-tumor expression. And I was not talking about this, but you can also look it up in our publication that was recently published in Nature Communications about the confident bioinformatical safety level. And we were able to derive functional CAR constructs in vitro and in vivo. So with this, I want to thank the people that really do the work, because I'm just sitting in the office and waiting for nice data. But this is really dedicated to the people in my team that compiled all the data, worked on this, and also a lot of Miltenyi colleagues, because it was a big interdisciplinary project with bioinformatics, our T-cell experts, and many others, but also cooperation partners in academia and also industry, and of course also you for your attention.
Thank you, Olaf, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, how do you remove the stainings between each cycle in the MACSima?
Yes, thank you very much. That's a very good question. So we're using 2 different technologies to do so. The one, and let's say that the first technology we applied is photo bleaching. So we are using dyes that are sensitive to bleaching, which are then fine for the imaging. But after each imaging cycle, we are then bleaching the dyes away. And we've also proven that even if you do a couple of hundred of stainings, that is still perfectly fine, and the tissue integrity is kept. And as a second removal mechanism, we are using enzymatic removal of the staining. And so we are using both systems in parallel. And also, if you perform one MACSima run, you can also use antibody conjugates, which are of the one or the other technology, and use it on the very same instrument in the same run, and just tell the instrument what removal to use in each cycle.
Thank you. Next question. How do you decide to culture primary tumor cells in 2D or 3D?
Yeah, so that was also, let's say, a surprise for us when we started about 5 or 6 years ago with the culturing of primary tumor cells, because the idea was that most epithelial tumor cells at least would grow in 2D, at least when you plate them on a substrate where they are happy to adhere. However, what we have seen is that in many patient samples, even epithelial tumors like pancreatic, ovarian, but also colon carcinoma samples, the cells like more to grow in 3D in tumor spheroids or organoids. And let's say that the challenging part is that you can not really see before from the patient sample, the diagnosis or the stage, if the cells tend to grow in 2D or 3D. So usually what we do is we dissociate the sample, isolate the tumor cells if needed, and then we plate them on primaria surface plates so that promote adhesion of the cells. And we have seen that if the tumor cells tend to aggregate and grow in 3D, they still do so when you have them on these kind of plates. So we do not do initial, let's say, a matrix gel embedding or so. And when we then observe during the first passages that the cells really like to grow better in 3D, then we usually switch to different surfaces, so such as ultra-low attachment plates, and then we culture them just in 3D. But yeah, it is very important to really keep it in mind that even if you culture epithelial tumor cells and your cells stay in the supernatant, that does not necessarily mean that they are dead, but they just can also grow in suspension. So it's definitely good to take these suspension cells, do a live dead cell dye staining and then check if these cells are still viable and maybe just tend to grow so. I mean, there are some particular tumors such as colorectal cancers where there's really a huge trend to grow them in 3D. And then what you can do also right from the beginning is to embed them in Matrigel. And the good thing is that the media that we developed are compatible with these 3D Matrigel embedded tumor spheroid, tumor organoid culture, as well as for the 2D culture.
Thank you. Next question. What if the enzymes used in the tumor dissociation step cleaved potential CAR targets?
Yeah, a very important point during the dissociation procedure that you have to worry about. As briefly talked about in the presentation, it's really a matter of using very purified or highly purified enzymes, because usually what causes the trouble is not the enzyme itself, but the residual activities that are still in the enzyme due to the manufacturing process. So most enzymes are manufactured from crude lysates, not recombinantly. And that means, for example, if you're using a collagenase and that still has a tryptic or chemotriptic residual activities, that will cleave off your targets because the collagenase itself is very specific. However, a certain amount of these activities is also beneficial for, in particular, tough tissues. And that's the reason why we have this strategy that we use combinations of enzymes that are each very pure. So we know exactly which activities, specific activities they have, but then we use a combination of these highly pure enzymes to still get a good dissociation and a high viability. But the advantage here is then now, of course, because you have these mixture of highly purified enzymes, that you do not have these lot-to-lot inconsistency. So you do not have to worry that one lot might degrade sensitive epitope, whereas the next lot has a different behavior. And that is really the key to do so. And how we check this is we do epitope stability tests on very sensitive systems such as PBMC. So we take cell lines or primary cells, blood cells that express these surface markers, and then we incubate them with the enzymes. And that is very sensitive because there's no extracellular matrix, so the enzymes can really act on the cell surface proteins. And by this, you then, by flow cytometry, we check if the mean fluorescence intensity, so the signal strength, is going down by the enzyme incubation in different titers. And by this, we can really narrow down what epitope sensitivities we are seeing in terms of certain epitopes and certain enzymes, and can tightly control this. And by this, you can be sure that you do not have a lot of false negatives, which you would have when you use either impure enzymes or very aggressive proteases such as trypsin or papain or disbase or something like that.
Okay, thank you. Next question. Are you planning on applying this method to identify targets in other types of tumors?
Yes. Yeah. So today I was talking about pancreatic adenocarcinoma, but we are also applying the very same method to other tumor indications, mostly, of course, solid tumor indications, because the workflow is really optimized for solid tumor indications. And I mean, our focus is really immunotherapy, so CAR targets. We are also working with TCRs. That was not a focus of today because the method of discovery is, of course, very different. One, you're using more NGS applications, but still for functionality testing, the very same workflows can be used, as well as, for example, for checkpoint inhibition, which is also applied to multiple tumor indications.
Okay. It looks like we have time for one more question. How did you select the initial antibody screening panels?
Yes. So basically, we have a screening plate. It's a plate. It's a cell surface screening panel, and basically it contains all the antibodies that are expressed on cell surface. We have it for human. And so it's a pre-selection of, let's say, of all the portfolio antibodies that are directed to work in such assays. And this is also constantly updated. So the portfolio is expanding, and therefore also these screening plates get larger. And what we then sometimes add, if they are not anyway in these screening plates, is, for example, targets that are known from other tumor indications and where antibodies that are amenable for either immunohistochemistry or flow cytometry in particular, as I showed today, are available. Then we add them to the plates.
Thank you, Olaf. Do you have any final comments for our audience?
Yeah, I mean, I would like to thank you all for, depending on your time zone, getting up very early or staying very long for the presentation today. And I hope you liked it and enjoyed it. And yeah, if any question comes up in the future, please just get in touch with us. Thanks a lot.
Thank you again, Olaf, for your time today and your presentation. We would also like to thank LabRoots and our sponsor, Miltenyi Biotec, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.